Okay, Saida, please start.
Good morning,
You can
everyone.
start. Yes.
Good morning, everyone. Good morning, sir, our president, sir, Mr. Desmukh, Dr. Professor Desmukh, sir. And also good morning to our today's safe speaker, Dr. Mosumi Devnath, ma'am. Ma'am, I welcome to our series of guest invitees. And I'm very happy to welcome all the participants, all the senior faculties from different universities. So firstly, I will like to start by introducing myself. Presently, I am a PhD research scholar in the Department of Microbiology from Assam Don Bosco University, Guwahati. And I'm also doing a job as a lecturer in Dispur Paramedical and Nursing Institute. And today I'd like to take one opportunity to give a news to you. Recently, 2 days before, I have been selected as a district microbiologist under Govt of Assam. So, sir, I actually wanted to call Desmukh sir for giving the news, but it's good today morning I got the opportunity in front of everyone. So I'm very glad to inform that. And without delaying, I would like to introduce our today's guest, very proficient, very expert from her field, Dr. Mosumi Devnath, ma'am, who is presently an associate professor in a very senior basis from Department of Biosciences, Manipal University, Jaipur. And if we look into the ma'am's qualification, she have done her MSc in botany with microbiology specialization, PhD in plant biotechnology, and also having lot many awards, including all-rounder awards. And she's having a postdoctoral experience, having an Endeavour postdoctoral fellow from Australia award, and also she have been having lot many award as Australian award visiting scientist to Purdue University, USA. And ma'am has a basic interest in biosurfactants. So today she'll be delivering a special lecture on biosurfactants and eco-friendly approach for remedial technology. So I hope everyone is eagerly waiting to hear ma'am. So without delaying, I will introduce ma'am to take the screen so that our participants will be benefited by ma'am's lectures. So ma'am, over to you.
Thank you, Saida. I'm privileged to Arvind sir to invite me for this prestigious lecture. Thank you so much, Deshmukh sir. I'll start my lecture. Is the screen visible?
Yes, sir. Yes, sir.
Yes.
So, Microbiology Society, I feel, is doing wonders in making microbiology a part of our life. People know microbes are present in and out of our body, but making this knowledge through the different students and the invited talks, I feel, sir, Microbiology Society is doing wonders. So I move into a topic that is biosurfactants and eco-friendly approach for remedial technology. As we all know that water, food, and shelter are the three major requirements of a human life. So this particular topic I have chosen because water is the major requirement of our life, and we need pure water that is potable and fit for drinking. So looking forward, I enter into a domain which is called as biosurfactants. They are basically surface active compounds with hydrophilic as well as lipophilic activities. Now, these compounds are found naturally in the microorganisms, and they can be utilized for different types of purposes. Here, I would like to distribute my work that I would be speaking today into the topics like what we have been doing with in the field of my biosurfactants, how our endeavor to the novel halophilic microorganisms started. And thus we started characterizing these halophilic biosurfactants. We worked on the oil recoveries as well as how these biosurfactants are effective to develop a novel moving bed biofilm reactor for the wastewater treatment, for the industrial purpose, as well as for the domestic wastewater. So this is Sambar Lake, and a unique place in Rajasthan, which shows the different colors due to the presence of large number of microbes present in this water. As you see here, this is the train which carries the salt that are naturally synthesized in this natural estuarine. This is the salt beds, as you see here. And we started working with this place for the exploration of certain microbes that can be very useful. So we made a whole track of it, how the different sites are there. And we started working. As you see, the water is not always at many places the same color as red. So we moved into isolation and characterization of the mud as well as the brine water from all the sites, as you see mentioned. And we collected a potential group of microbes. And these microbes could do a function in certain types, like they could decrease the surface tension, as well as they could create certain type of emulsification, frothing. And what we saw was the surface tension decrease happened not only on the after 24 hours, but it also decreased after 48 and 72 hours respectively. So the emulsification index was also taken up, and there was another test that we found out that was very useful in screening, that was the hemolysis. So we did find certain clear zones. So we did the initial understanding of why more amount of bacillus was found there. So the gram-positive bacteria were more in these cases, which were potential biosurfactant producers. And we came to an understanding that there was a specific gene which was called as the surfactant synthetase gene. And there was an NRPS gene also, the non-ribosomal peptide synthetase gene. And these was present in all the bacteria, specifically the bacillus group of organisms. So this NRPS gene was not only the one, but we also later on found out that there was another type which was responsible, and that was not the NRPS gene. So that was responsible for the rhamnolipid production. So as you see here, we did the understanding that this surfactant molecule has a similarity with all the molecules which were isolated from the bacillus. And then we went for characterizing this surfactant. As you know, surfactant is ones which is mostly present from bacillus. So we tried to characterize it, and we found out that through the liquid chromatography mass spectrometry, that almost all of them coincided. The chromatograms, the TICs that we found, as well as in the MRMs also, we did find that there were quite a big similarity, where the initial adduct found was something around 1036. It broke down into the parents to form 685. And this molecular weight was, this was a deuteryne. And we were very much clarified that these molecules were nothing but, as everybody has reported, it is the surfactant. So we also wanted to look into how did they look through the FESCM, and we found out that the major parameters were carbon, oxygen, and as because it was from the salt prone area, the salt tolerance was also about 10 to 12% in most of the bacteria. So we found sodium was also there. Thereabout, we also looked for the fatty acid methylated esters, and a similarity of the TICs were recognized, and we were confirmed that now that most of the people, whatever they have told till now about surfactants, it was clear that this was a surfactant, and then we went for the isolation, the extraction, and the characterization. So the extraction for our molecules, chloroform and methanol was the best type. And this surfactant could really be used in future as a part of the remedial technology. But most oftenly, when we look into any industry, oil is also a part of the many processing units. So we wanted that actually, can oil be also be recovered by the use of these surfactants? And we found out that yes, sufficient amount of oil can also be recovered as you look into this figure. And we also found out that when they are compared with certain standards, the standards gave us that they were working quite similarly, and in some cases they were even better. So the isolation and characterization of more bacteria continued, and we came across another one, which was Streptococcus. It was Streptococcus syzuri. This was also found to have a copper translocating P type of ATPase. So we moved into it, and we found that this type can be easily used for other activities like heavy metal chelations and so on. This is another type of a biosurfactant. This was identified by Rhodococcus. This Rhodococcus ruber, which we isolated from, again, a salt-tolerant type of, this is a salt-tolerant bacteria. This was not a halophilic. We isolated it from the effluent, textile effluents. As you find here, the extraction was again the same, and we could easily identify it when extracted through chloroform methanol. So in most of our cases, we have been able to identify it with chloroform methanol, and these molecules were very good in decolorization of the bacteria. So as you see, we have done the decolorization with the industrial chemicals that are used in different forms, as well as with natural colors also. And in all of them, we could find the good decolorization effect. So we started moving into the fact that mixed microbial cultures would be much better. And this was a test where we did with single cultures as well as with mixed cultures. As you see here, the mixed cultures are some of them which had a better effect. So we looked into the different types of theories which could be answered for the mechanism, and we came across basically 4 different mechanisms which can explain this. So it might be due to the cell surface adsorption, as you find here. It can be by transformation of metals into the non-toxic form. So they can also do this by oxidation or reduction. They can also be something which we call it as the efflux movement by the metal transporters which are present on this cell membrane. And there can be certain intracellular accumulation through the special components. Again, these are some of the transporters present there. There may be a metallothionein which are working here, as well as there can be bio-removal of the metals through the biosurfactants. So with the micelle formation, they can be precipitated out of the complexes. We did find the different types of antimicrobial activity because mostly biosurfactants are said to be non-toxic. So we did use them with different standards against the organism which was producing, as well as we also did it through different types of on the non-pathogenic, although we have used, but the different types of other organisms also. So this was some of the activities that we did with our isolated biosurfactants, and we did find good activities. So I move across to move into my main domain, that is the remedial technology. And going forward, I find that the scenario of today is full of industries with the growing impact of urbanization. So what do we find? We find that there are a large number of pollutants, and these pollutants, which are just trying to create toxic molecules in our body also, because some or other way it is coming into the food chain. So the pollutants range from dyes, the heavy metals, the plastics, the polyaromatic hydrocarbons, the pesticides. So these are the major types of the pollutants which come from the from the industries. Apart from that, we also have the other molecules which are also present in the sewage. So in many places, we find that the industrial effluent is going and mixing with the somewhere through the sewage also. So in the sewage, we found out that not only there is a high amount of BOD, but there another thing happens which are in the nano pollutant form. Those are the antibiotics. These are the antibiotic residues which basically we are having antibiotics, and that is going again into the influence. So we need to work on this particular aspect, how to make water potable or make water at least ready for a particular agricultural use. So looking to this fact, we started working using our set of biosurfactants. So we found out that the existing treatment which happens would range from adsorption, membrane filtration, oxidation, ozonation. There are use of Fenton's reagent, photocatalysis, activated sludge system, sequential aerobic and anaerobic system, fixed bed, their coagulation, flocculation. But we thought that can we give something which is going to remove the chemical oxygen demand? There's a nitrification. The aesthetic value relates to how you see the water. That is, we need to remove the color. We need to make it free from any odor. We want to remove the most important thing, that is the heavy metal, that we need to remove it. So taking to this fact, we started moving to the area of wastewater treatment. This is one of the plants in Maharashtra only. And when I asked to send them some of their pictures, they send me these pictures, and if you look into it, it shows that organization of a wastewater treatment has not been planned properly. And just looking into the fact that there are so many such tanks. So these tanks, if you look, they are not properly oriented. Now let's see how basically it looks like. So this is the existing tanks in operation, where I don't find this is the collection tank 1, collection tank 2. This is the neutralization tank. I think so. There's a background noise. There's a background noise. Can we mute? Can you hear me, everybody? Can you hear me?
Yes, ma'am.
Yes, ma'am. Now it's okay.
So if there are so many tanks, number one, there'll be a lot of energy that would be required to run all these tanks. Second, number two, think about the space which will be utilized in putting all these tanks. So space and energy is the two most important things that an industry needs to look into. So if they keep on spending everything on their electricity, how will they generate positivity towards the society? So they'll be just wasting the energy. Looking into it, so the problem that these industries told me, the number one is the color removal, the odor issues, and this was persistent in textile, herbal, nutraceutical, the food industries, and the most important thing is the effluent treatment is very slow. So it takes a long time to make the water reuse, and they needed large number of space. So the clean water often doesn't meet to the statutory pollution control norms, and there were many other industries who were continuously seeing that the CPCB or the pollution control board authorities are coming and they are just sealing their industries because of the effect of the industries to the neighboring villages or the neighborhoods. So there's always a constant fear from shutdown. So our technology started for these people, to the society, and to provide them clean water. And we started using the same consortia of our biosurfactants for the clean water. And when we made a product, some people who
পরিমাণে হ্যাং হয় কি বলবো মা ফোন আসছে ফোন দেখতে পাচ্ছি না তারপর মা এখন যে চালাচ্ছি
So these type of biosurfactants, can we use it as a mix to fasten the process of the biological treatment? So this mix would eliminate the odor, the color, and some of the other parameters which are essential statutory norms for the water treatment. And thereby, the reduction of the sludge was another thing which was bothering all the industries. So can we make a reduction in the sludge, thereby save money by reducing all the operation cost? This is such a small graphical record of what we have done. So we used a consortia along with the biosurfactants, and these biosurfactants will allow to grow certain type of flocks. And these flocks that you see, the biofilms, these biofilms are going to eat away all the organic matter, and this will decrease the BOD and the COD too. So this is the effluent inlet. The primary screening will be done by coagulation flocculation if required. We have done many such treatments where only there is a single operation tank, and this operation tank runs through a aerator. The aerator moves through a system, and then finally it is the settling tank. If there is requirement, then we can have a tank too, which will be again a storage, and whatever minimal amount of sludge would be there, it would be collected. This water is 100% used for agriculture till now in most of the places wherever we have worked. The global market of MBBR as such is something around 6.2 billions, which is going to be one of the prime method, because this is a safe energy as well as this is the one which is going to save money. So both ways there is an economy to space as well as to energy. And there is a revision which says that with a large number of other microbes coming into the water, this type of a system must be the best method. I cannot say is one best method. It can be one of the best method which can really help in recycling of water. So the market, we are not just the one who is using this type of an MBBR technology. As you see in the industrial market and in the national market also, there are certain participants, and hence the competition is there. So if I look into the product, I find that the requirement are small plastic floaters, which are called as the carriers. And these carriers are we cannot say they are recyclable. They are the ones, if you just inoculate, if you just allow them to grow the biofilms, they are sustainable. So the cost and the maintenance is also near to negligible. In this particular aspect, we have already published one patent, and we have also used this biosurfactants to make a detergent additive. And lately, our design patent has also been, design IPR has also been filed just 2 days back, based on a combinatorial method, which includes MBBR and a method called as hydrocavitation. So in this particular aspect, I have received 3 proposed projects also, and this has helped me to become one of the person who can help in reducing the pollutants in industrial water as well as in domestic water. So moving bed biofilm technology is a technology where I'm using these different types of biocarriers. As you see, there's a huge technology in building up this carriers too. As you see, the architecture of these carriers are different. So they are basically polypropylene made structures, and they range from a very minimal size to a very complicated type of a design too. So in the market, you will find 2 basic colors available. One is white and the other one is black. Usually people offer these black ones because they are much more effective, but they have one type of an issue, that is they leaked out this black color. But this type of a technology is the one which can guarantee you to give you less sludge, less energy consumption, and less space usage. So what do we find? That these are the ones which literally keeps on growing all this small microorganism inside the cavities, and these are called as the carriers. So they carry the microbial load, and these microbial load basically eats away all the pollutants. So the high density pollutants are finally taken off by these carriers. I have also given a list of this different carriers. As you see here, they are of different architectures, and lot of researches are going on only in designing this particular type of the biofilm carriers. Basically, they are floaters, and they have a density that is similar to that of water. Just to revise for people who came just now, who have joined in, so I talk about biosurfactants, which are surface active compounds produced by microorganisms that can efficiently remove all the organic pollutants, which also includes, as I have shown you in my previous slides, oil also. So they are having the least toxicity, and they are biodegradable in nature. So I talk about my pollutants, my biosurfactants, which are available on our lab-based extraction methodology from either halophilic organisms or halotolerant organisms. So these extremophilic biosurfactants have a ability to function at extreme salinity. So they have a low critical missile concentration and they are having a good bioavailability. This is the cleansing activity that we have also found out that these biosurfactants producing organisms are having large amount of enzymes, and these enzymes are basically, we focused on two basic enzymes. One is protease and the other one is lipase. So this is how we have done some of the immobilization experiments too. Later on, we can discuss. So this is our innovative low-cost wastewater treatment technology, where we are focusing on all the industries as well as the domestic sewage. We look forward for an innovative technology where there's only one-time inoculum charging. One-time inoculum charging is basically a step-up method that we are going for. And this step-up method is utilizing a consortia of halophilic and halotolerant microorganism, as well as the biosurfactants isolated from, extracted from these organisms. We have the biocarriers, which are going to work in decreasing the the pollutant level. We are having a specialized design tank for the efficient biodegradation, as well as this is one of the economical solution for wastewater treatment. So we have found out that the nitrogen has been reduced to 92%, the BOD 75%, and the COD 85.2%, which acknowledges the fact that the water can be reused. So it is a continuous process. I repeat, this process has least maintenance cost. So this is a continuous process, and it doesn't involve those so many tanks that we were talking about. So it involves only one tank, and we can have a customized service for each industry. Thereby, the most problematic thing, which is always in hype for wastewater treatment, is does it remove the heavy metals? So cadmium, chromium, lead, nickel, arsenic. We found out that most of these can easily be removed. In case of colored waters, especially in our state of Rajasthan, we have around 2200 plus industries which are making dyes or using dyes. So we have a lot of issues with colored water, which also contains toxic, excuse me. This also contains the toxic molecules, and these toxic molecules are containing lot of heavy metal. So the application of the biosurfactant in the removal of heavy metal is one of the most important requirement of wastewater treatment, and our molecules, our biosurfactants, are working wonders in it. So our study started with this lab, this industry, as I told, in Maharashtra. We were working for an IGSTC project, and this is on a herbal effluent that we started working. As you find that one alone, this strain could easily decolorize the water. We started working with basically two different types of carriers. One was the K1 and the K3. And after a due process of step up, we could find that 7 days was minimal requirement for the removal of the different pollutants, and the lead also was reduced. So we initially started with 5 liters, and later on we moved to around 200 liters. And I think so you can find that the type of frothing that was created was very much efficient in promoting the process at a very high scale. And this was one of the demerits I mentioned with the black color one, the carriers. But if you just centrifuge this in the lab to just note that whether it's working or not, you can see the difference. It's absolutely clear water. So as I told, all the molecules, this is the copper part where colored water is there. There is always a probability of having copper. So all the molecules were able to, our mix composed of surfactant and rhamnolipid basically. So we were able to reduce this heavy metals to a larger extent. And as you know, lead is one of the most heavy metal which is talked about in terms of wastewater, and we were very happy to see that the lead amount also decreased. The molecules alone also, the molecules I keep on saying because the biosurfactants from the different organisms we were using, as well as we were also using mix of these surfactants and the rhamnolipids, and we find that this consortia was better. So we started working with only the consortia later on, instead of moving through the single cultures. And in most of our experiments, we found that after upgradation, it takes around 20 to 25 days for upscaling the process. It is 7 days which gives us the result. So these are all the results as you find. If I'm doing it in a very proper way, and if the, in fact, the microorganism don't behave same with all the different types of, this is an experiment which we did with herbal effluent. We moved from the lab scale to the pilot scale. But if I look into it, always it is not the same time that we need. So in some cases, we found out that it was 72 hours also, which was quite efficient in reducing, but it is not always the same. So you can see in one of our experiments, we find that we went till 15 days and then we were satisfied that the organism is working. So the organism and the biofilm that is formed, it takes a certain period of time. The organism also needs to grow in that effluent water, and the effluent water where it acclimatizes, then only it starts working. So that part we are looking forward is the upscaling. That part depends on how much amount is the pollution load and how much will the microorganism able to adapt to it, along with the biosurfactants, which creates the foaming because the aerator is moving continuously and the system works. So to a certain point, we were able to work with it, and then we moved into another industry, which was the ceramic industry. The ceramic industry also, we did some of the lab scale, then we went to the pilot scale. Now we started moving with, as you see here, we have the white color biocarriers and we also had the black color. When we went for an upper, we found out that when you are going for around 20 liters, this type of biocarrier was apt. But looking to the cost efficacy, when you're going up to 200 liters or for 3 KLD, we have done with 3 KLD also in this system. So it was economic to go for the black color carriers. In all of them, we found out these carriers, this is the system where we have worked, and this system allowed us to give them, because here the major issue was the chemical oxygen demand. So we could create such a situation for this industry that now the industry doesn't even require to put the coagulants in the very first tank. So without that, initially they were putting the coagulants. So now they don't need the coagulants. Without the coagulants, the consortia and the biosurfactant mix is able to work on it. These are all the results. As you find, there's always a reaction which took us some time. And this is, as we are showing you, it's a result of 72 hours. When after 20 days, this is after the 20th day when the stepwise acclimatization has been done, we found that the TDS, pH, EC all have decreased. And there was a subsequent reduction in the COD also. These are the COD values. And this is on the on-site treatment. It took us a long time to reduce. So these are around 9 days it has taken. But it again depends on the type of effluent that we are working. This is with the lead. So lead, we were able to reduce it, but this reaction is still continuing. So we are not able to give you the exact data because lead, we need to go for around 0.01 mg per liter. So that is still yet to get. So this is the type of biofilm which has been formed. I think so now you can understand this is one different type and this is the other type. This type will allow more organisms to accumulate. So this is a type which is called as the K1 and this is the K3. And this is from our ceramic industry part. We have tried to isolate the microorganisms. We have tried to scrape out the biofilm and we found out that they are almost same as that of the surfactant. Lately we have done some work with the sewage treatment also. This is one of the sewage treatment plants which we have at Manipal University, Jaipur. So one of my student, she started working on this. The basic thing was we wanted to check whether MBBR with our novel treatment can make a change in the concentration of the different pollutants, as well as can we make a change in the total nitrogen content. So Geldal nitrogen was done, and we were also concerned with the BOD. In the earlier ones, we were concerned with the decolorization. The second one in the ceramic, we were concerned with the COD, and here we were more concerned with the BOD. So this is the sewage treatment that we started working. We have still not gone for the main tank, that is the 5 KLDs that we have right now. But our experiments has been quite fruitful when we have done till the 20 liters. As you see here, we have upgraded the system, adding a large number of other molecules. And till now, we have got some of the biofilm which has been attached here. So we found out that there was a increase in the TDS and the EC. But if I look into the TSS, the total suspended solids, we find that after a treatment of 20 days, the parameters has decreased. The same happened with MLSS. MLSS, we found out that this mixed liquid suspended solids was according to the norms that we wanted. The COD was reduced. As you see, it has come till 400. There was something around 393 or something. So after 25 days, this is our reading that we got it. Similarly, the BOD, this is what our main focus was. We did find a comparative requirement which most other technologies are doing, because MBR technology is also working side by side in another tanks. And we found out that our requirement was to use our MBBR technology along with our biosurfactants, and the treatment was observed. So with the Geldal also, Geldal nitrogen also, we found out we got a better result in it. Sorry, no. So the conclusion to this sewage treatment was that we found out reduction of around, in case of BOD, there was a reduction of 77%, which can be a way towards achieving our target. So we were basically in this experiment, we were basically focused on BOD, as well as we were focused on this total Geldal nitrogen. So we got a denitrification, and the rate of reduction was around 38%. Here we have used only one type of a carrier, and we are proposing to use a few more carriers. Sometimes mixed carriers also help. So we might be going for K1 and K3 carriers too. We promote this technology through Biosolve Innovations Private Limited. It's a startup founded by me, and we have been doing some of the other activities by moving through technotrons also. And we have 1 patent and 2 trademark to its credit. Thanks to my team of PhD scholars and postgraduate students who have been working in this particular efforts to make biosurfactants as a molecule of unique applications. And thank you all for patient listening. Over to you, sir. Over to you, sir. Deshmukh, sir. Thank you so much. Sayada, ma'am. Hello.
Yes, ma'am.
Hello.
Now we'll check if someone or somebody have any question. So, any participants, any questions?
Yes, very good morning, ma'am. Very nice presentation and very nice information you have shared with us. Ma'am, I have 2-3 queries. Like you mentioned that you're using carriers Q1 and Q2. Can you say, can you define please what do you mean by Q1 and Q2?
It's K1 and K3 that I said.
Okay.
Yeah. Yeah. They are certain carriers. They are basically floaters.
Okay.
I think so. You must have seen, I have explained them. They are certain carriers where they are made of polypropylene. See, these are the ones. Can you see?
Yes, ma'am. Yes,
So,
it is visible.
yeah, yeah. So there are different types of carriers. I also showed you some more carriers. See, they are different architecture they have. And these carriers are the ones in which these molecules, these microorganisms come and they settle there. Okay. And they start, so it is not just bacteria who settles there. In the due course of time, you can find some of the other organisms also coming and settling there. So basically, these are the carriers which will develop a particular type of a biofilm. And hence, this methodology is called as moving bed biofilm reactor. Okay. So this is the ones which is moving. So in the water, because there is a aerator there. Yes. So we have a motor and a diffuser at the bottom of this tank. So these tanks, what would happen is the aerator will continuously move the water, right? Yes. And this particular carriers will be going again from bottom to up, and they will be revolving all throughout. And during this process, they will be interacting with the pollutants. When they interact with the pollutants, and a biofilm is constantly being formed in this biocarrier, so what will happen? Some of them will go and stick to it, right? Yes. The pollutants, and they will start reacting with it. So in the due course of time, they will again break it down, because as I said in my first part of my talk, that how biosurfactants work. Yes. So they'll break down all the pollutants and it will settle down at the bottom. So what you get basically is a pure water, and a water which can at least be used for irrigating your plants.
Okay. Thank you, ma'am. Thank you for sharing the answer. Now, my second question is, as you said that you are using consortia. So in that consortia, you are focusing only biosurfactant producing organism, or you are focusing on a bacteria that will degrade the effluent, and after some time you are adding a biosurfactant in that?
No. We started our work with halophilic organisms, right? And these halophilic organisms are all biosurfactant producers from Sambar Lake.
Okay.
So we got around 16 very potential group of biosurfactant producing organisms, which were basically synthesizing two molecules. One was surfactant and the other one was rhamnolipid. So
Okay.
these molecules had the potential. Then we wanted that our efficacy of the system should grow. So we moved to textile effluent contaminated soil. And in textile effluent contaminated soil, again, those organisms are halotolerant, right? We checked it, tested it. So now our consortia is unique and novel group of biosurfactant producing organism that synthesizes surfactant and rhamnolipid as a part of their system. So we have taken the extracted biosurfactants as well as this mix of mixed microbial culture to make a unique combination and treat this wastewater.
Okay, ma'am. Okay. But how you are going to confirm that when the organisms are utilizing effluent, they will only produce biosurfactant, because it is quite possible they are going to release some other metabolites, and which can be one of the reason for reducing the chemicals which are present in the effluent.
See here it is. How can I kickstart my reactions? Okay. So when I'm putting it in a bulk of a industrial wastewater or a domestic wastewater, the major thing is at that time there is huge amount of pollutants. So creating a system of cleansing has to be done with the cleanser, right?
Yes, yes.
So biosurfactant here instigates the process. It is not the one which is continuously been created. In due course of time, I'm not taking out my biocarriers. It is present here only. And it is at that time the molecules biosurfactant has started the process, but it is at that time the biofilm where the residents of the other organisms have started. But initially it is simple nylon or polypropylene or plastic floaters. So how can I think that my microorganisms will grow there? So this biosurfactant starts the process and initiates them to form the biofilm. Okay.
Okay, yes. Oh, thank you so much, ma'am, for answering. I would like to share that I'm also working on a biosurfactant from last couple of years. I have noted your contact details. I will be contacting you soon, ma'am.
Sure. Anytime.
Yeah, thank you, ma'am. Thank you for giving such a relevant information on biosurfactants. Thank you, ma'am.
Thank you so much.
Hello.
Yeah, please.
Yeah, am I audible, madam?
Yes, you are.
Yeah. Good morning, ma'am. Thanks for this informative session. Even I am also working on a biosurfactant for the remediation of the oil oil and heavy metal contaminated environment. My question is that, ma'am, we check the hemolytic activity, okay, of biosurfactant producer. So, what is the relationship in this biosurfactant and checking the hemolytic activity?
Yeah. See, when I speak about hemolytic activity, then it is the blood. Blood is what? It contains some amount of protein also, right? Heme is the protein, iron and globin is the protein, hemoglobin, right? So when I look for the blood content, if it is able to break down certain type of protein, maybe this is one of the screening. I'm not very sure, but I think their activity to break down the protein is also unique, which has been found. Maybe. I'm not very sure of the answer. To break down the protein. That is the only thing that it is doing. See, if I'm looking into we are using in different proteases activity also. So there also it's a complex molecule that it is breaking down. And that's where you see a clear zone, isn't it? So maybe this biosurfactant producing organism has the ability to break down complex molecules. Maybe this is the
ma'am.
answer,
Yeah.
but I'm not very sure. Yeah.
Okay, ma'am. Thank you.
Yes. Thank you.
Good morning, ma'am.
Good morning.
Ma'am, I'm audible to you?
Yes, yes. Please tell me.
Ma'am, actually, I'm an MSc student and my dissertation work is on MBBR. Now, what
Okay,
I've done is I am optimizing the model. I've almost optimized the whole lab scale model of MBBR. I'm right now doing the continuous model.
Ah, great.
Ma'am, what I faced was when I optimized, as I was doing optimization, I had used 3 different types of biocarriers.
Oh,
One was Calden's 4, and another 2 were one of that you have shown, the black one, and another one was small black one.
okay, okay.
Now,
So K3 and the K1. It's
problem
K3
is
and
that,
K1. Uh-huh.
the problem was that I used it. As we had only 6 months for dissertation, I cannot go for each and every parameter. The parameter that I focused was COD, BOD, TSS, MLSS. I hadn't gone for nitrification, denitrification. Now, what I observed was the biofilm was not forming on the black ones.
Okay. Yeah, I showed you. It takes some time to form on the black, but I do have some very good results on black one also.
My white ones were giving so good results.
Can you see?
But
Can you see?
Ma'am, I had the same kinds of result. The result that decolorized one you are having, the white one were giving same results. The odor was totally gone. There was the SVI index was also much lesser in the continuous one. But I don't know why the black ones, the biofilm are not forming on them. They are polypropylene, but the reason I am not understanding it.
Yeah. So what you can do is you can use what we have done. We actually made a mix. Did you go for upgradation of your system?
Yeah, ma'am. First, I did the flask level one. Then I had gone for the batch reactor. I had the 10 liters of reactor.
No, we don't do it in this way. What we do is like we go for upgradation of the system. See, your bacteria won't grow when you start putting it directly into a 3 KLD or a 5 KLD or a 10 KLD, right? So you increase that volume little by little. So this is upgradation. If you do the upgradation and then put it, this is just a result of 7 days. This happened very fast. That the one, if you can see
the results you are showing, the same results I have got in 10 liters of reactor with white ones. The results were tremendous. The odor was totally gone.
Okay. But with black also it happens. When you are going with biosurfactant and microorganism mix, you can get the same thing with that also. Okay, you can try it out with this, right? But remember, you have to go for upscaling the process for the bigger volume of water too. Okay. You don't put, let's say you are trying to inoculate 1 liter in 5 liters. Don't do it in this way. You go little by little, and then add the bulk into the next level. Understood?
Okay, ma'am.
And you start putting your carriers on the day 1 only. Don't put it after you find that there is some amount of decrease in your parameters. Don't do it that way. Usually people
No, ma'am. What I did was the very first day only I put the effluent and the carriers at the same time, and the inoculation also. But this was some reasons that I was getting the results wrong, that the black carriers were not forming. If there is no biofilm formation, then I cannot upgrade it to continuous one, because in continuous we don't need inoculum.
Yeah. So you can add, did you extract the biosurfactant?
No, ma'am. Actually, my work was not on biosurfactant.
If you see here, See, the work actually initiates with biosurfactant. It doesn't initiate with the microorganisms as such. It also, here our whole technique is with biosurfactants. If you look here, if there is a cleansing activity, if you look at the type of foaming that you find here, so the microorganisms will grow. The system, in many cases, it so happens if there is more foaming, some of the biofilm also, it starts eroding out. Okay. But in due course of time, you also find that after 15-20 days, even in the black one, I don't have those figures right now, but you do find good amount of biofilm there also. What is your source of effluent?
It was a effluent from CETP plant near Palsana in Gujarat.
Okay. Okay.
As it was CETP, I couldn't go for a very specific one because every time I collect the effluent, there was some different kind of effluent coming. So that was also a problem. And another problem was that
basic problem with the effluent treatment, like you never get the same type of pollutants every time. So you have to change your strategy, something or the other.
Okay, ma'am. Thank you. And also, ma'am, I have done the SCM because it was an optimization model. So what we did was we performed the scanning electron microscopy of both the white one and the black one. But from that results also, we obtained that the white one was showing good amount, bulky amount of biofilm, while the black ones, there was only flat and dry type of biofilm formed.
Yeah, Khushbu, I would like to tell you something. See, I just showed you a range of biocarriers, right? Why did this range of biocarriers start? Because in some of them, the biofilm formation is less. And in some of them, the biofilm formation is more. Okay. If you see the K3 and the white one, see the number of apertures that they have. Maybe these are the ones which are better and hence people are using this type, right? Instead of using the K1, which is only small. So maybe the amount of biofilm residence time too is less in this K1 where the number of grooves are less. That is the reason why still people are doing a lot of research just to make these carriers. Okay.
Okay, ma'am.
Understood.
Yes, ma'am.
So people are doing a lot of research in this, making the biofilms, they carriers also. So maybe you can take up this subject and do with the type of knowledge that you have gained in your dissertation. But nice work, nice discussion.
Thank you, ma'am.
Thank you.
Ma'am, I have one question, ma'am. Can you please?
Yes, yes. Please.
Yes,
tell me.
have you tried, ma'am, with fungus also? Is because fungus is also working as a, like they're also reducing the like effluents load or organisms. So is there any work like, ma'am, have you seen like fungal? Can be the fungus will be used as a like biosurfactant, something like that? Because I saw the bioabsorption of some pathogenic organisms. The fungus will also clear them. So is there any like anything which, because I want to apply, I'm looking for a person that
I understand your work must be related to fungus, right? I did my PhD with Albugo candida, but I have not worked much with fungus after that. So I cannot work with fungus and bacteria simultaneously. So we restricted to extremophiles and the extremophilic bacteria. But yes, there are a lot of literatures where you can find that fungus is also doing the same type of an activity. I'm not very much sure of the extremophilic fungus or not. I have not seen, but I have read some of the papers where fungus is also working quite well. You can try them, but try them with extracted biosurfactant and your fungal cultures together, you will get better results and faster results. Okay. Anyone else?
Ma'am, we have one written question. The question is
Yeah.
how you come to know the all microbes uses consortia utilizing the pollutants.
Okay, we have done a few experiments where I have shown you where the microbes were tested on, see, this was one. If you see, we have tested with all of them alone also. And this was a single application, and then we have done the mix. Now, when we did the single application, we did get like SE1 was very good. I'll show you. Yeah, this is the one. This was very good. So what we did was we later on tried to find out that how these organisms are working in a group. But as I told you, my main focus was on the biosurfactants, not the biosurfactant producing organism. My biosurfactant and biosurfactant producing organism, the main job was to create a faster pace in forming a biofilm. So the mechanism was to make a biofilm, but the time taken to make this biofilm was a huge time. So I reduced the time using this consortia of biofilm. So here my thing was, if you remember, I showed you a large number of bacillus group which were making the surfactant. And I think so I missed one. There are a few types, like Rhodococcus is making both surfactant and rhamnolipid. We have a Pseudomonas, which is also making again the similar type of rhamnolipid. We have another molecule, Enterococci. So that was also making rhamnolipid. So we had something around a group of 20 bacteria which are making surfactant type of biosurfactants, and we have a small group of biosurfactant producing microorganisms which are rhamnolipid. So what I did was in order to understand that actually is this due to biosurfactant or not, we scraped out the dried biofilm that we could procure from the carriers, and those we confirmed it through the FTIRs, and we did find it that they had a large amount of rhamnolipid as well as surfactant. So that gave us a clarity that maybe in due course of time, it is the biosurfactants which were working to reduce the pollutant level.
Ma'am, in continuation to the question, I just want to know how you are so confidently saying that organism is only producing rhamnolipid, because whatever the nutrition we are giving to the organism, as my understanding is, so they can produce different type of biosurfactants also.
Yeah. We did test. We found out the maximum amount was surfactant and rhamnolipid. We have done the FTIR only for this particular case, but we got good results with the LCMS also. So we have tested that maximum amount was with surfactant and rhamnolipid. That we could get it. Yeah.
Okay. Thank you so much, ma'am.
Yes. Any other questions?
Ma'am, another question from the inbox.
Yeah, please.
I'm a second year BSc student. My team is going to research on biosurfactants. So can you please suggest which surfactant give the good result?
I didn't get it. What they want from me?
Which surfactant give the good result? Yeah, they need a suggestion from you. BSc student.
Yeah. Actually, I don't understand what they want, but see, biosurfactant is a molecule which is generated in a bacteria. Okay. So first thing that you have to do is you will have to grow this biosurfactants in a specified media. We do it with MSM and we add 1% crude oil to it. And then we incubate it for a certain period of time. We usually do it for 7 days. And then we continuously try to give the same type of treatment. By the time we know that those specific genes have now been upregulated, we know that these molecules will be synthesized in due course of time in a large number. And thereby, we take those organisms which are now pure cultures, we develop them into pure cultures, and then we start our work for this was a primary screening, then we take the secondary screening. So it will depend upon what type of organisms you are getting from the base matrix from where you have isolated. Is it clear? So then you will have to check two major things that will give you an understanding that your biosurfactant is actually potential. The first one would be your emulsification values, and the second one would be how good they are able to reduce the surface tension. We don't even consider the hemolysis as one of the most effective way to judge whether it's a biosurfactant producer or not, but we do consider whether it is able to reduce the surface tension. So if it is able to reduce, that means it was doing good amount of work. It may be a good biosurfactant producer in due course of time. And the second thing is whether it's making frothing, it is responsible for frothing. And if so, what is the E24 value, that is emulsification after 24 hours. And then you can subsequently do it till 96 hours and then find out how much amount that it is generating. So what was the level that you are getting with the oil and your basic supernatant that you have extracted, right? If you do this, two things, you will understand how potential your biosurfactant is. And then you have to proceed for any type of an application that you want to do. Is the answer making happy for you? Yes? Hello?
Yes, ma'am. It was a very informative answer for them. Ma'am, I think we have one more question. Yes.
Yeah.
How far this biosurfactant will be effective for heavy metal remediation, madam? I am a PhD on bioremediation on toxic heavy metals. So can I apply this biosurfactant for remediation of toxic heavy metals?
Yeah, sure. See, when we speak about heavy metals, we are basically concerned with 5 of them: lead, arsenic, mercury, cadmium, chromium, right? So as I showed you in one of my slides, that these molecules, this is already a published paper. So this is what can you see my screen? See my screen? You see my screen?
Yes, ma'am, it's visible.
Yeah. So this is what we are proposing. And you would also check that there are basically 5 mechanisms in all the literatures. They are saying that the biosurfactant is able to reduce, or they are able to convert it into a non-toxic form. So these are the 2 major things that they are saying. The reduction may be usually by converting it to non-toxic form. And we have found that reduction is there in almost all the different types of effluents we have worked with. So if you are biosurfactant producer, that's what I always say, use chromatographic methods to understand the potential of your biosurfactant. Like we have done LCMS strip and quadrupole, you isolate your biosurfactant and then you quantify it. Now we have a consortia of biosurfactant producing organisms with different levels of biosurfactant production that we have found. So either you have to choose optimization as one of the route to enhance your biosurfactant production, and then use that biosurfactant in your experiments and check the usage of it in reduction of your pollutant. So it all happens when the molecule is a high biosurfactant producer. It is not just the organism alone can do it. You have to check the solid form of the biosurfactant also. So we have used the solid form. In some cases, the bacteria produces a large quantum of biosurfactant. But if you choose a consortia, totally they are producing a small amount of biosurfactant, then the time required for the reduction of the heavy metal or the quantum of the heavy metal that they would act upon would be less. So the product, the final result is you get a very less reduction in your heavy metal, which you wanted, you did not get it. I hope you get me. Is it clear? We are not just talking about the biosurfactant producing microorganisms alone. We are also talking about the indigenous production which has been extracted out and characterized. So you use both of them together, the biosurfactant producers and the biosurfactant extracted. Is it clear?
Yes.
Am I understood?
Okay, ma'am. Okay. Very good.
Yeah,
So
thank you.
now I request Tamanna, please conclude it, because if there are more questions,
Yeah,
personally they can ask to ma'am.
thank you. Thank you.
Yes, yes. And really, I'm thankful to you, ma'am.
Thank you, sir.
I know you are an active faculty from Rajasthan
Thank
and
you, sir.
continuously doing many things for the students.
Thank you,
That
sir.
is a good part from your side. You are always fighting for betterment of the student.
Thank you, sir.
So we want your cooperation in the future for the development of microbiologist society.
Sure, sir. Sure, sir. Anytime.
Yes. Yes, sure, sure. Tamanna,
Thank you so much.
please go ahead.
Thanks a lot, ma'am, for such a great, beautiful session. It reflects
sir.
how much expert you are in the biosurfactant is actually a very huge topic, but ma'am has explained it in a very simple words with every details, even giving lots of reference from our research work from very basic level to an advanced level, with giving examples, showing types, the pictures, the process of extractions, how technology started, solution to the drawbacks, even applying it in wastewater, which is one of the most important matter of discussion nowadays, and how biosurfactants are important to our day-to-day life. So we're also happy to see how ma'am has done patents and project related to the topic. So it's a step toward research and development. So thank you so much, ma'am, for making our Sunday a very fruitful one. We are very grateful to have you, ma'am. And also thanks to all the participants who have attended today's lecture and making the session more complete. So over to you, sir.
Thank you. Thank you very much. Thanks all for attendees also. And thank you everyone. This lecture series will be continued. Tamanna is very active, so I have given responsibility to her. Again on next Sunday, there will be one more lecture. Thank you.
Thank you
Thank
so
you.
much, sir. Thank you so much. Thank you, Tamanna ma'am.
Thank you, sir.
Okay.
Thank you, ma'am.
Thank you.